Welcome back to the diagnosis part. As I said, there will be three parts. Now we are in the second part. So how we will be able to diagnose the bacteria. So in case of E. coli, as we know, there are four types. So you will be able to understand every mechanisms because I have explained shigellosis, the mechanism, so it will be more similar. So I will explain the very first mechanism. So the first is enterotoxigenic E. coli. So this E. coli is known as travelers bacteria because the persons which is traveling more and eating more roadside food, they are most likely to be infected by E. tech. Now there are the virus. So as I have said, like E. coli and Salmonella has type 3 secretion. So here you can see the like needle like processes of the bacteria, and they're injecting toxins directly, and inside the cell there are heat labile exotoxins and heat stable exotoxins. So heat stable exotoxins bind to the cell membrane, and this will activate the guanylate cyclase, and then it will also convert GTP to the cGMP, and it also phosphorylate the ion channels. So the ion channels will be able to like open, and as a result from the cells, sodium and chloride and water will be able to go out of the cell, and it will cause the watery diarrhea. And here you will be able to like read the mechanism, and this is the same as this like heat stable mechanism. So we need to remember that this E-tech, E aggravative E. coli, and E pathogenic E. coli, these three types of E. coli causes watery diarrhea. So this is really important, just remember. And the enterohemorrhagic and E enteroinvasive E. coli, this causes bloody diarrhea. So we know from the names that hemorrhagic, it is must that it will cause bleeding. And the EIEC, which is enteroinvasive E. coli, it is most likely be more invasive, and it will cause also blood secretion. Then let's come to the diagnosis of the E. coli. So to diagnose the E. coli, we need urine, feces, blood, and sputum, and pus, and CSF. So you need to remember one thing, that in our body, blood and CSF, which is in our spinal cord inside it, cerebrospinal fluid, these two components, liquids, are most sterile. So we don't need to cultivate them or do something else, and we don't need to keep it in the refrigerator. So just take the sample from the patient's body and directly pass it to the lab and do the microscopic and also biochemical properties identification. So we don't need to cultivate them. Then you just simply read it. Then we need to like transport the sample we took it from the patient. So we need to remember that we need to transport the sample as soon as possible, like within 2 hours, because if we use the simple container, that so the sample will be safe for 2 hours, and the temperature must be 2 to 8 degrees, and also 24 hours. So I'm sorry, I confused it. So in the container we will be able to contain it for 24 hours, but we need to transport it within 2 hours. So if the patient is more severe, so we need to transport it as soon as possible. Then this is the cultivation time, which is 37 degrees Celsius and 24 to 48 hours. We need, actually, this is the optimum time bacteria can grow. And here are the agars, McConkey agars, Levin and endo agar. So if the agars change from red color, like if the agar is, for example, pink, it changes to red, so there is must some kind of fermentation occurring in the agar. And we need to I need to give important information that nowadays this endo agar is not used that much because it is kind of old-fashioned agar, so we don't use it in the lab. Then there is blood agar, then the bismuth sulfite agar, Kliger's iron. So the Kliger's iron, we need to know one information, that we don't use this agar to cultivate we don't use it for cultivation, we use it to know the features of the bacteria. So if the bacteria is glucose fermenting or the lactose fermenting or not, or H2S producing, hydrogen sulfate producing or not. So these are the features of the bacteria. To know the features of the bacteria, we use this agar, but not to cultivate it. To cultivate the bacteria, we use it McConkey agar, Levine agar, blood agar, bismuth sulfite agar, and also semi-solid agar is used to see the motility of the bacteria. So as I've said, like if the bacteria has one fimbria, it's more motile. If the bacteria has multiple fimbria or fimbria, then it's not that motile. Then for the identification, we need to go step by step. That is, we need to know we need to do the microscopy, then we need to do the biochemical properties to know what kind of antigen does it has. Then we need to know the sero identification, we need to identify the antibodies. So if you Google it, you will be able to know more, or from the uni you will be able to know. You already know, or you already studied this process, agglutination or latex agglutination. Or if you want to know more, just like say in the comment section, do I need to do any kind of video about these processes? So I will do. Then the fourth step is bacterial susceptibility to the antibiotics. So in this test, we actually want to identify that what kind of antibiotics it can invade the bacteria. So these are the three options we use in the lab, which is E-test, disc diffusion test, and also Vitek 2. And if the patient is more severe, so to identify the bacteria right away, we use the PCR to know the gene which is actually producing the toxin, and also ELISA test. So just do let me know, do I need to do a video about these processes or identification processes, then I will do. Then in the Salmonella Diagnostics, we know here we need to take the feces because it's related to the diarrhea. So, and also it causes the typhoid fever. And then we have the transportation, Carrie Blair, and specifically for the blood, we need to take it in a back take. It is a modern bottle, like it's a glass shape, like it is made of glass, so it's more modern. And here are the cultivation. These are the most likely same thing, because this Salmonella Shigella goes under the Enterobacteriaceae. And there is the identification, same process again, same thing, just only one thing is important, that it doesn't Salmonella, it doesn't ferment the lactose, except it doesn't ferment the lactose. and we need to proceed like E. coli diagnostics, because I've said again that these are more same. And then the Shigella diagnostics, same thing. Just read through this diagnostics table and you will be able to get it. And again, Shigella, the one thing you need to remember, that does not ferment lactose except as Shigella sonnei. It slowly ferment lactose within after 48 hours. Slowly ferment lactose, and it takes 48 hours. So the same identification processes. And here the yersinia. So for the yersinia, we need to take feces, urine, pus, and also biopsy from the lymph nodes. And then we need to we can carry it through the sterile container, carry bleh, and then port a cool, which is a plastic shape bottle. We can keep the bacteria in that. And again, backtake for the blood, only for the blood. So we cannot use we cannot use any of this for the blood. No for blood. If we want to use for blood, it should be backtake, only backtake. Just keep that in your mind. And here again, it does not ferment, yersinia does not ferment lactose. So this is the, again, identification is the same. So this is the diagnostic process. Let's end it here. Let's go to the clinic.